In this video, we're going to try and pull together much of the information you've covered so far about the innate immune response by looking more closely at the process of inflammation, which will allow you to put what you have learned so far into temporal and spatial context. As an overview of some of the things that we will be talking about in this video, and hopefully many of these will already be familiar to you, this diagram summarises quite nicely the processes that are involved in the inflammatory response. So we will discuss the recruitment of cells to the site of inflammation, the removal of apoptotic or infected cells, and the repair process that occurs. The first thing to consider is what inflammation is. When someone says inflammation to you, what is it that you think of? What image springs to mind? And I'd imagine many of you from first-hand experience are thinking about a red and swollen joint or a painful knock. But from an immunological perspective, inflammation is defined as a non-specific, localized but complex biological response to tissue injury. And these sources of injury can be varied, including pathogens such as bacteria, fungi or viruses, damaged cells caused by physical injury such as a cut or other irritants, including chemicals or radiation. And the physiological response involves the blood vessels, cells of the immune system, and the molecular mediators that these components release. Now, the reason that it is important that we discuss inflammation as a whole is that it is, as a process, implicated in a huge range of health complications, from neurological to cardiovascular to autoimmune diseases. So in order to understand what is going wrong in these scenarios, you need to really understand what should happen under normal circumstances. Under normal circumstances, the role of inflammation is to eliminate the initial cause of the injury, to then remove any cells or areas of tissue that have become damaged or necrotic, and subsequently to initiate and regulate the repair and recovery process. And this is what that looks like. You will all have suffered a cut or burn, for example, and over time that injury is repaired so that you would never know that the area had been damaged. That means that we need to think of inflammation as a beneficial process. It is not harmful to the body under normal conditions. However, in the case of infection, the process of inflammation has to be tightly regulated, because what is harmful to the microbe can also be potentially harmful to the body. You may have noticed that I have been very careful about my choice of words when describing either inflammation or infection. And the reason for this is that the two terms do not mean the same thing. Inflammation is not a synonym for infection. And the reason for this is that, as we defined earlier, inflammation only involves the immunovasculature, so the cells of the immune system, and blood vessels, irrespective of the cause of that inflammation. In contrast, infection describes the interaction between the inflammatory response and the invading pathogen. And it's really important to remember this distinction because many inflammatory situations do not involve a pathogen, so they're not driven by infection. As an example, hypersensitivity reactions, where there is an inappropriate response to a normally harmless substance, or atherosclerosis, where the inflammation is due to the deposits within the blood vessels surrounding the heart. Unless you've been living your life in a bubble wrapped up in cotton wool, then you should all be familiar with the cardinal signs of inflammation, without actually having stopped to define them. These cardinal signs were first described by Celsus sometime between 30 BC and 38 AD, and described the localized reaction to injury or infection. These cardinal signs were defined as rubor, tumor, calor, and dolor, referring to the redness, swelling, heat, and pain associated with the inflammatory response. These signs can easily be seen in this image of a patient's ankle and foot. But what caused this? Do you think it was inflammation or infection? Well, in this case, it was an inflammatory response, as all this swelling and redness that you can clearly see, along with the heat and the pain that I'm sure you can imagine, was caused by an allergic reaction. Somewhere between 129 and 210 AD, Galen added a fifth cardinal sign, which was functio laesae, so the loss of function associated with the other cardinal signs. One way to remember these signs is through the acronym PRISH. So pain, redness, immobility or loss of function, swelling and heat. And these are also illustrated in this cartoon. So hopefully between the photo of the patient's ankle, the acronym and this cartoon, you will be able to remember these cardinal signs quite easily. It's also important to remember that inflammation is a beneficial process to the body under normal circumstances. These cardinal signs can be explained by the physiological responses that occur in the body during inflammation. The redness and heat are due to the vasodilation causing increased blood flow nearer the surface of the skin. So this brings blood from the centre of the body closer to the surface, which explains the warmth associated with inflammation. The swelling is also due to vasodilation, but in this case it's the leaking of fluid from the blood vessels into the tissue which carries mediators of inflammation to the site of injury or infection. Whilst the pain is due to the release of chemical mediators, including histamine and bradykinin from resident cells such as mast cells, and these all combine in different ways to contribute to the loss of function. So the area might be too swollen or too painful to move, for example. So now that you have an idea of what inflammation is, let's discuss what causes the acute inflammatory response. So this is the normal, beneficial inflammation associated with repair and recovery. Inflammation is initiated by tissue-resident immune cells, for example macrophages, dendritic cells, and mast cells. These cells are able to interact with pattern recognition molecules, both membrane-bound and soluble, which have been discussed in more depth in previous videos, but which can be broadly divided into PAMPs, which are pathogen-associated, or DAMPs, which are linked to tissue damage. These interactions result in the release of inflammatory mediators, for example histamine and bradykinin, that contribute to the onset of the clinical cardinal signs we have already discussed, with distinct vascular and cellular responses. Looking specifically at those vascular and cellular responses, we can link the vascular components to some of the cardinal signs and then to the subsequent cellular processes. So the vasodilation and increased permeability account for the rubor, calor, and tumor, so redness, heat, and swelling. The reduction of local blood volume due to the permeabilization results in an increased local concentration of red blood cells, a slowing of blood flow, and this allows for the recruitment of leukocytes from the circulation to the tissue via a coordinated sequence of events which we will describe in more detail shortly. And this is what you can see in the diagram here. On the left is a healthy prostate gland and an inflamed prostate gland on the right. And what this looks like in tissues in terms of cellular recruitment can be clearly seen in the differences between the sections of the images, where in the inflamed tissue, each of the dark purple areas is a cell that has been recruited into the inflammatory site. There are a range of inflammatory modulators that contribute to the vasodilation and permeability of blood vessels. The vasoactive amines such as histamine and eicosanoids including prostaglandin E2 and leukotriene B4 are released from macrophages and mast cells, whilst nitric oxide is produced by macrophages and endothelial cells. The overall net result of the release of these mediators is to remodel the local vasculature, allowing the movement of protein-rich fluid into the tissue, which carries amongst other things components of the complement system to the inflammatory side. Furthermore, if the inflammatory stimulus is a wound, then additional elements including platelets and coagulation cascade factors will also be recruited. So let's take a simplified overview of this response to start with and revisit this diagram. In the first image, you can see a cut has created an opening in the skin and has introduced pathogens into the wound. So in this example, we are going to be talking about infection. The damage to host cells, coupled with pathogen-associated molecules, triggers the release of chemical signals like histamine, leukotrienes, or prostaglandins from the tissue-resident mast cells and macrophages. These chemical mediators stimulate the endothelial cells of the blood vessel, and you can see the vasodilation in images 2 and 3, with gaps starting to form between the endothelial cells to allow for the movement of phagocytic cells from the circulation into the tissue to help remove the pathogens and any damaged cells. In the final image, as the infection is brought under control and the wound starts to heal, homeostasis is restored, vasodilation subsides, and the phagocytes will undergo apoptosis and be removed from the tissue. So you can appreciate there is an awful lot going on here, and there needs to be a coordinated sequence of events. The recruitment of inflammatory cells from the circulation is a key factor in the resolution of inflammation, so we will look at this next. One of the key functions of vascular permeabilization as a result of inflammation is to induce blood stasis. That is the slowing down of blood flow, which allows leukocytes, and in particular neutrophils, to be rapidly recruited to the inflammatory site. This recruitment follows a coordinated series of events that make up a process called extravasation, and we will look at each of these steps in sequence. So as an overview, before we look at the molecular basis of extravasation, we can describe a sequence of 5 steps, which starts with the marginization of leukocytes, so that is the movement to the edge of the blood vessels. Once at the margins, the leukocytes will start to roll along the vessel walls, which has been activated by the chemical mediators from the inflammatory stimulus, resulting in adhesion of the leukocytes to the endothelium. The leukocytes flatten themselves and then undergo a process called diapedesis or transmigration. And once in the tissues, the leukocytes will follow the chemotactic gradient to the site of injury or infection. So we can go a bit deeper into these processes now that you have a broader overview. And the first stage is marginization. Under normal flow, the leukocytes are confined to the axial flow of the vessel. But blood stasis, due to permeabilization, pushes the leukocytes to the margins where they will roll along the endothelium, forming a pavement of neutrophils along the endothelial walls of the vessels. As they start to slow down, they adhere. So this cartoon shows an overview of this whole process, and we'll break it down each step. The process is initiated by the release of cytokines like TNF-alpha and interleukin-1 beta that have already been discussed, and these are released by activated tissue-resident macrophages and mast cells and act on the vascular endothelium. These cytokines will upregulate the expression of E-selectin, whilst P-selectin is expressed on endothelial cells that have been activated by histamine, complement component C5a, leukotriene B4, or bacterial cell wall components like LPS. L-selectin can become upregulated on activated leukocytes and will help bind to proteoglycans on the endothelium, whilst a special glycoprotein on the surface of the leukocytes called Salil-Lewis X modified glycoprotein can interact with P and E selectins, and it's a combination of these interactions that will slow the leukocyte and allow it to roll along the vessel wall, which is what you can see summarized here in the first part of the diagram. A combination of this rolling action and chemokines acting on the leukocytes convert another family of receptors on the surface of the leukocyte called integrins, which include LFA1, MAC1, and BLA1, from a low to a high affinity state. And once this happens, the integrins can bind to their ligands on the endothelial cells, which include ICAM1 and VCAM1, which are intercellular and vascular cellular adhesion molecule 1. This allows for firm adhesion and rolling arrest, with the binding forces being strong enough to resist the shear force of the blood flow. This arrest allows the leukocytes to spread along the endothelium, and this will allow for the next stage of the process, which is transmigration. The transmigration, or diapedesis, occurs through a combination of cytoskeleton rearrangement, forcing a pseudopodia into gaps between the endothelial cells, and the action of platelet endothelial cell adhesion molecule, or PCAM1, which you may also see called CD31, which is present on both the endothelial cell and the leukocyte, acts a bit like the hooks and loops of Velcro to pull the cell through the gap. And once inside the tissue, the leukocyte will follow a chemotactic gradient to the source of inflammation. To highlight just how important this process is, we can look at a condition called leukocyte adhesion deficiency, or LAD. LAD was first recognised as a pathology in the 1970s, and 3 types have been identified, which are imaginatively named LAD 1, 2, and 3. It is quite a rare genetic disorder that occurs in approximately 1 in a million births, and there is no racial or ethnical predilection. The hallmarks of this deficiency are delayed sloughing of the umbilical cord and recurrent bacterial infections during infancy, which is underpinned by defects in the neutrophil adhesion stage of extravasation. The molecular defect responsible for this deficiency is caused by an autosomal recessive mutation in the ITGB2 gene, which encodes for the beta-2 subunit of integrins. As a result, neutrophils are unable to arrest because the integrin cannot bind its ligand, and therefore diapedesis cannot occur. The neutrophils are unable to reach a site of infection, and bacterial expansion proceeds largely unchecked. So essentially, the inability of neutrophils to arrest means that they become trapped in the blood vessels, are unable to reach the site of bacterial infection. Under normal conditions, however, we will see the resolution of inflammation, which is the eradication of the threat, be that tissue damage or infection, and the restoration of tissue homeostasis. Just as inflammation is actively induced, it is actively terminated. Just as there are pro-inflammatory cytokines, there are anti-inflammatory cytokines, and these can be released to terminate the inflammatory process. This is aided by the short half-life of the pro-inflammatory mediators, the removal of inflammatory cells via apoptosis, and an active switch to anti-inflammatory cytokines and chemokines production. The main reason for doing this is to protect surrounding tissues, because the cells involved in the inflammatory response, particularly neutrophils and macrophages, are capable of massive damage to our own tissues. The acute resolution of inflammation can be associated with an acute phase response. You may remember from a previous video, we briefly discussed acute phase proteins, and these form part of this response. So the individual may suffer from fever, which is a temperature above 37.8 degrees Celsius. And actually, this elevated temperature is beneficial for the immune system, as it impedes pathogen growth or replication, but enhances the immune response at the same time. This fever can be accompanied by elevated heart rate and blood pressure, chills, and anorexia or loss of appetite. If the inflammation is due to infection, then different pathogens will drive different immune responses. So bacterial infection is characterized by elevated neutrophil levels, viral infections by lymphocytes, and in the case of parasites, eosinophils, which can also be elevated during an allergic response. We also see the production of some of the positive acute phase proteins by the liver, so we will see elevated C-reactive protein levels, or CRP, and increased erythrocyte sedimentation rates, or ESR. Now, as I'm sure you are aware, the resolution of inflammation does not always go perfectly. And there are essentially 4 different outcomes to the inflammatory response. The best case scenario is the restoration of homeostasis with no complications, but in some cases we may see pus formation in an abscess, and there are varying degrees of severity here, as anyone who has watched the Dr. Pimple Popper video on YouTube will attest to. We may also see fibrosis, which is the technical name for scarring. So if the wound is large, or there is a large abscess formation, we may see scarring. And again, this can be mild or it can be severe, and in bad cases can lead to a loss of function for an area. In the worst case scenario, we progress from acute to chronic inflammation, and this can have life-threatening consequences. And this diagram illustrates these 4 different outcomes and the progression between each. Note that it is possible to go from chronic inflammation to fibrosis, but the extent of scarring and the extent of loss of function is likely to be much more extensive than other routes to this outcome. So what exactly is chronic inflammation, and what can go wrong? Well, at a basic level, it is the difference between healing and destruction. The acute phase response, as we have discussed, can rid the body of infections or repair the damage due to irritants, cuts, burns, or trauma. But chronic inflammation results in pathologies including cancer, autoimmune diseases, neurological diseases, or cardiovascular diseases. If we compare and contrast acute and chronic inflammation, we can see distinct differences, ranging from the speed of onset to the duration of action, the types of cells that are involved, and the potential outcomes. So the 2 processes, although they start from the same place, ultimately result in hugely different outcomes. Acute inflammation is rapid and short-lived, involving the recruitment of neutrophils, as we have already discussed, which subsequently undergo apoptosis to resolve inflammation. Chronic inflammation, on the other hand, takes a long time to develop, but is persistent, and there is a shift away from polymorphonuclear cells to mononuclear cells, such as monocytes and lymphocytes. Necrosis further enhances the inflammatory situation and leads to fibrosis or scarring, and the formation of new blood vessels as even more immune cells try to infiltrate the area. Inflammation is a huge global health problem, so much so that back in 2004, Time magazine ran a cover article calling inflammation the secret killer. And whilst inflammation is normally self-limiting, in chronic inflammatory situations, this process is persistent and can be driven by a range of causes, so persistent injury in the case of ulcers or infection with TB, inhalation and subsequent prolonged exposure to toxic agents like silica or asbestos, or if the immune system starts attacking self-tissue, we see autoimmune diseases develop like rheumatoid arthritis or lupus. To revisit this diagram from earlier in the lecture, inflammation can play a role in all of these different pathologies, so it can impact on pretty much every system in the body with devastating and debilitating consequences. So that is an overview of acute and chronic inflammation. What I've tried to do in this video is draw together many of the concepts that we have covered so far in the other videos. I hope that you found this useful and it has helped you to provide a better picture of how the various components of the innate immune system come together to help defend our body from a threat, whether that is inflammatory or infectious. That pretty much brings us to the end of the innate immune system, although we will revisit some elements of this system as we move forward and start looking at the highly specific adaptive immune system. Because remember, these two arms of the immune response are always working in parallel.